Hello. Last class we have started introduction to biomedical engineering, and some of the cases we have seen that biopotential has been generated from different organs, different organelles, and also from cellular level. So that is why we must know about the basic cell structure and their functionality of the organelles which is present inside the cell. And nowadays this field also provided by different courses in post graduation level or graduation level in IITs and different reputed institutes. So somebody who can create some interest in this field, they can also pursue further as a higher study. So we must know the basics about the cell structure. Today in this lecture we will learn some about that. So now basically we know that the cell is the fundamental unit of any living organism. So some of the cell working together for a particular goal, we call them tissue, and some tissue will functioning for and creating one organ, then some organ and organ system will create the organism. So in this manner cell plays one vital role for the activity or for a living organism. So we call that as a fundamental unit of life, which is essentially development for existence of organism. So basically 30 to 40 trillion cells are present in a human body, and all these cells they form some of the organs. Cell contains hereditary materials, and they also which also helps us to produce the cell division and reproduction. Cell contains cytoplasm, genetic material like DNA and ribonucleic acid, then proteins and other biomolecules. And the study is technically this science or the branch we study about the structure, function and nature of the cell is known as cytology. Basically the cell term comes from the cellular. Cellular means small compartments or rooms. It is one Latin word. So now we'll see what is cell theory. Basically the cell theory is proposed initially by two scientists, Sladen and Swan. It is proposed by two scientists. Then one is Sladen and Swan, and in 1830 they says that all living things are made up of cells, and cells are the smallest working unit of living thing. So these two has been first proposed by these two scientists, Sladen and Swan. Now additionally the Virchow will give the third term or third postulate, that is all cells arise from pre-existing cells through cell division. So this is most important, and the third one has been proposed by this scientist Virchow. So we'll move to the next slide and see some of the major components like cell membrane and cytoplasm and how they behave. So cell membrane, basically the animal cell, or they are made up of phospholipid or bilayer of phospholipids. You mark here, there are two layers. One is this one, you mark red color ball like things, red color heads, and two yellow color strands on both sides. This is one layer and second layer is just here, and this is your outer side of the cell. We call it extracellular fluid, and inner side we have cytoplasm. In between them, this cell membrane separates this extracellular fluid from cytoplasm, and throughout this or across this cell membrane you will find some of the proteins embedded inside it, some of the channels, protein channels, which which are selectively permeable and make this membrane selectively permeable, which ion or which particular component will enter into or out of the cell. It is decided by these channels, and this membrane is also embedded with globular proteins, surface proteins or peripheral proteins, then glycolipids, and also there are many alpha helix proteins. This phospholipid layers, they are this red color ball like thing, they are actually called hydrophilic heads, and the strands are hydrophobic tails. So these two are present in both the layers, upper and lower layers. So cell membrane is a structure, you can mark here, that gives rigidity and protection to the cell. It controls the movement of the biomolecule in and out of the cell. It also protects the cell and its organelles from the external environment. By nature, the cell membrane is semi-permeable or selectively permeable, which we will discuss later on, and how it helps in developing the cell potential, that also will discuss in the subsequent lectures. So now we will see another important component of the cell is the cytoplasm. Cytoplasm, you mark, this is one anatomy of a cell. Here you mark that the cell contains or different organelles are present inside this cell, and they are just floating in jelly like structure, which is known as your cytoplasm. And inside this cell, all the biochemical processes or reactions are going on, like glycolysis in the cytoplasm itself. All other organelles are suspended into the cytoplasm of the cell, and the fluid thing or the fluid portion of the cytoplasm is known as cytosol, which contains water, dissolved ions and small molecule except all other organelles. So one by one we will see what are the different organelles. We mark here the very clear picture. It is, you can mark in this picture, you will see different organelles. First one, the central part here, this one, this is your nucleus. The central part of any cell is nucleus, and this nucleus is again the center of this nucleus is known as nucleolus, and nucleolus surrounded by nucleoplasm. That fluid which encloses or which is surrounding that nucleus is known as nucleoplasm, and then it has been enclosed within the nuclear envelope. But this nuclear envelope has some nuclear pores certain pores are there, through which exchanges of nutrients, exchange of materials or biomolecules takes place. Now we have different organelles like mitochondria, we have centromeres with cytoplasm, lysosomes, ribosomes, rough endoplasmic and smooth endoplasmic reticulums. Then we have Golgi apparatus. One by one we will discuss all these important organelles and their functionality. As we have discussed, the nucleus, it is a double stranded organelle, double membrane organelle that present in eukaryotic cells, because as we know we have two different types of cells, primitive cells like prokaryotic and the which is present cells which are eukaryotic cells. Eukaryotic cells, all the membranes are we have all the nucleus are double membrane organelles. and contains most of the cell's genetic material. You mark here, genetic material like DNA and in form of chromosome is present inside the nucleus. The nuclear envelope consists of inner and outer phospholipid bilayer membranes, as in case of cell membrane, which separates nuclear content from the cytoplasm and is studded with nuclear pores, that is your nuclear pore, through which the transportation of molecules takes place. These nuclear pores are large protein complexes, or you can mark, as we have seen in case of our cell membrane. Here also we have protein complexes or protein channels are there, which regulates the selective exchange of proteins, RNA and micromolecules between the nucleus or nucleoplasm and cytoplasm. The outer nuclear membrane is continuous with endoplasmic reticulum. It is another organelle, and this nuclear membrane is extended and attached to this endoplasmic reticulum, and over this endoplasmic reticulum we have ribosomes attached, because these ribosomes, we will see, these ribosomes are helpful in protein synthesis. We will see that. The nuclear lamina, there are mesh work which is or filaments of proteins, that is lamins, which is present inside the nuclear lamina, which is called nuclear lamina, lines the inner membrane and provides the structural support and help organize chromatins. Chromatins are those they are actually the hereditary materials, or it is related to chromosomes, and this genetic materials has been organized and present inside this nucleolus, and which has been protected or organized by or supported by a structural work of nuclear lamina, which is made up of your lamins. So, nucleoplasm, as we have discussed, it is also similar to your cytoplasm. It is gel-like matrix inside the nucleus. It contains different types of enzymes, ions, nucleotides and nuclear protein, which are helpful for DNA replication and transcription. This is the most important thing you can say for the. So, next, chromatin is the complex of DNA and it associated with protein, mainly histones. What happens? This histone, they created a octamers, which is just acting as one core, and surrounding that the DNA strands has been, they are just bounding that octamers, and in this manner they create these chromatins, which remains dispersed during interphase and condensed to form visible chromosomes during cell division. So, they are very crucial for cell division and carrying out the hereditary characters. So, we have 23 pairs, human have with 23 pairs of chromosomes. Out of that, 22 pairs are known as autosomes, which are basically they are responsible for different types of characters developed in a human being, and one pair out of this 23 are called as sex chromosomes, which decide whether the person or that baby will be a female or male. So, chromosome is made up of single DNA molecule that tightly coiled around a histone protein form a compact structure within the nucleus. As you can mark, it is your nucleus. Inside that nucleus we have nucleolus. In the nucleolus we have these chromosomes, and these chromosomes, if you expand this chromosome, you can mark here there are 2 strands of chromosomes are there, and within these 2 strands you will find they are attached at a particular point. We call it as a centromere, and here they are, if you open that, we will find these are the structures which are the, we call that DNA structures, which are bounding or tightly coiled around these histone octameres and form nucleosome. Then if you open that, you will find all the gene structure, and these nucleotides are base pairs are guanine, cytosine, adenine and thymine. So these are the 4 difference which are present in a DNA. So chromosomes arms are one we have short and Q is a long, you mark here, this is your P and this is your Q arm. They are attached at the centromere as we have discussed, and during cell division they separate from this centromere. Then telomeres are the specialized DNA protein structure at chromosome ends. So that means this portion, these end portions, they are known as telomeres. These telomeres protect the DNA, they are protecting the DNA from deterioration and fusion. So chromosomes are classified according to the placement of this centromere to different or like metacentric, submetacentric and acrocentric chromosomes, but in also telocentric, but when this chromosome or the sister chromatids are attached to this two telomeres, then we can say it as a telocentric chromosome, but in human being no telocentric chromosomes are found. The nucleosome, the basic unit of chromatin, consists of about 146 base pair of DNA wound around a histone octamere, and this allows dense packing of DNA in the nucleus. And during cell division, this sister chromatin joint at the centromere separate during mitosis and meiosis, as we know during cell division. So then next one is your mitochondria, which is known as powerhouse of the cell, which generates the required power and by aerobic oxidation or respiration, you can mark, and specifically via oxidative phosphorylation on inner membrane of the inner membrane proteins. So we'll see how this mitochondria help in production of energy through this oxidative phosphorylation. Actually, mitochondria are they are double membrane rod shaped organelles. Typically their diameter lies between 0.5 to 1 micrometer. The outer membrane is a smooth and contains porins, means through that we have some it is the protein channels, we can say. Some of the permeable, they are permeable to small molecules, but inner membrane is highly folded into structures called cristae. So increased surface area for adenosine triphosphate production, means ATP production, is select, and this inner membrane is selectively permeable. So the mitochondrial matrix enclosed by the inner membrane contains enzymes. So as we can see, we have two membranes. One is outer membrane, which is permeable and it contains some porins. Similarly, inner membrane, which has a folded structure called cristae, they are selectively permeable. In between that, there is a matrix. In between this inner and outer membrane, there is a matrix, and that contains, those matrix contains the enzyme for the Krebs cycle. Krebs cycle is required for energy extraction through metabolism. So, and also it contains mitochondrial DNA, ribosomes and other molecules required for the mitochondrial function, enabling semi-autonomous replication and protein synthesis. Means this mitochondria is also one self-sustained organelles. It has its own DNA, it has its own ribosome for protein synthesis, and also all these enzymes for creation or extraction of energy. So this is the mitochondrial number and crystal density vary by cell type. You know, wherever there is a more requirement of energy, that it must vary accordingly, reflecting the energy demand. Muscle cells requires more energy, so that is why muscle cells have numerous mitochondria with extensive cristae. Now here you can mark the structure of this mitochondria, where you can clearly visible the mitochondrial DNA. This is your cristae. This structure is, these are the folded inner foldings are known as cristae, and this is your smooth outer membrane, and in between them you have all this matrix. This matrix will have, they are containing all the required enzyme or this mitochondrial DNA, ribosomes, everything is present in intermembrane space. So it is your outer membrane space. This functionality of mitochondria, you can say it produces energy. That is why it is known as powerhouse of the cell. It also helps or it also involved in another process that is called cell death or regulated cell death. So that is known as apoptosis. They regulate the programmed cell death by releasing cytochrome C and activating enzymes like caspases, essential control for cell destruction. So that means every time whenever the cell which is not performing well, that means they has to be disposed, means that is why we require that process is known as apoptosis, and new cells will be generated by cell division. So during that process, this mitochondria will activate will also help in activating the enzymes like caspases. It also stores calcium. In mitochondria absorbs and stores calcium ion, assisting in cellular signaling, muscle contraction and blood clotting. And another important functionality of mitochondria is thermogenesis, means heat production. In certain cells such as brown fat, mitochondria releases energy as heat to maintain body temperature. So this process is known as thermogenesis. Now next important organelle is your endoplasmic reticulum. As we have discussed, it can be of two types. So this endoplasmic reticulum is a network of membrane bound tubules and sacs known as cisternae continuous with nuclear envelope. If this is the nucleus and this is your nuclear envelope which has nuclear pores, then it is, you mark here, it is attached, it is continuous with this nuclear envelope. These endoplasmic reticulums are originated or attached from this nuclear envelope, and they are of two types. One is rough endoplasmic reticulum and another one is smooth endoplasmic reticulum. In the rough endoplasmic reticulum, you find these beaded structures, these starts with ribosomes. These beaded structures are ribosomes, the responsible mainly for protein synthesis and folding and quality control. These three are most important functionality of rough endoplasmic. They provides the support for attachment of the ribosomes which are helping in protein synthesis. Then smooth endoplasmic reticulum, it lacks ribosomes. In this side we will find these are the smooth endoplasmic reticulums. They involves in lipid synthesis. So you mark, once again I am repeating, rough endoplasmic reticulums which are studded with ribosomes, they are helping in protein synthesis, and smooth endoplasmic reticulums which lacks of ribosomes, they helps in or involved in lipid synthesis and steroid hormone production, carbohydrate metabolism and detoxification. So these are the important functionality of smooth and rough endoplasmic reticulum. The rough endoplasmic reticulum provides a large surface area due to the folding nature for ribosomes synthesizing membrane bound and secretory proteins that are folded and modified within the endoplasmic reticulum lumen. And proteins synthesized in raw endoplasmic reticulum are then packaged into vesicles. Nearby this endoplasmic reticulum, we have the vesicles which carries those proteins synthesized here and transport that to the Golgi apparatus. What is Golgi apparatus? We will learn later in the next slides. So, the smooth endoplasmic reticulum synthesizes phospholipids and cholesterol, essentially cell membranes. This cholesterol actually it is also required. So, they are also generated or synthesized in the smooth endoplasmic reticulum and produces steroid hormones in specialized cells. Smooth endoplasmic reticulum participate in glycogen breakdown. We know that it is called glycogenolysis and detoxification of drugs and poisons by enzymatic processes. Basically, these processes, this glycogenolysis and detoxification of drugs, all these processes are held in the liver cells. The endoplasmic reticulum acts as a storage site for calcium, crucial for signaling in muscle contraction and other cell processes. The endoplasmic reticulum helps maintain structural support and intercellular transport by providing membrane to organelles such as Golgi apparatus and nuclear envelope. That means these endoplasmic reticulums, they captures these proteins which has been generated at that site, they can be transferred to the Golgi apparatus. The endoplasmic reticulum membrane contains enzyme involved in various metabolic and synthetic activities that includes cytochromes and ATPase. So, these two are important for energy production and these are required in the mitochondria to generate energy. Sarcoplasmic reticulum, which is actually present in a muscle cell. Sarcoplasmic reticulum is a specialized smooth endoplasmic reticulum. They are present in muscle cells and it regulates the calcium ion critical for muscle function. So, in this way you will find different functionality of this endoplasmic reticulum. Now, come to this ribosome, which are also known as protein factory. Protein factory means they helps in synthesizing proteins. Ribosomes are complex molecular machines found in all living cells and synthesizes protein by translating mRNA or messenger RNA sequence into polypeptide chains. So, these ribosomes are composed of two subunits. One is one is large, another one is small. Both are made up of ribosomal RNA or rRNA and proteins. So, the smaller subunit binds the mRNA and decodes the genetic information. Smaller one binds smaller subunit binds mRNA and decodes the genetic information. But the larger subunit, it catalyzes the information of peptide bonds between amino acids, because you know that amino acids are the building block of peptide bonds, and peptide bonds, after create a long peptide bond, we can say it is a protein. So, ribosomes can exist free in the cytoplasm or be bound to the raw endoplasmic reticulum depending on the protein being synthesized. It depends which type of protein is being synthesized. The two subunits come together, means the longer and smaller subunits come together during protein synthesis, but can dissociate afterwards and can be reused. Only during protein synthesis they come together. Ribosomes have three main sites for tRNA. So, that is your binding during translation, that is site or amino acyl P site or peptidyl and E site or exit, which facilitate the stepwise addition of either amino acid or growing peptides. Ribosomes translate the genetic code from mRNA, or mRNA means your messenger RNA, in sets of 3 nucleotides called codons, each corresponding to a specific amino acid carried by the tRNA. So, now coming to another important organelle, that is your Golgi body. Here you can say this Golgi apparatus or Golgi bodies, they are this is the structure you mark. These are folded tube like structures you mark here. It is membrane bound organelle and composed of a series of flattened stacked pouches called cisternae, and typically they are 4 to 8 number in each animal cell or every animal cell. It is located near to the nucleus and often adjacent to the ear in the endoplasmic reticulum to the in the cytoplasm. Why? Because we have seen in the previous slide that these endoplasmic reticulums or rough endoplasmic reticulums, they are the site of attachment of these ribosomes, which synthesize the protein. Once the protein has been synthesized, that has been entered, that has to be transported from different site to different sites, and it can be done through this Golgi bodies. The Golgi body has structural polarity with distinct regions. 3 different regions are there: the CIS phase or cis phase, medial cisternae and trans phase. So, we mark this phase which is close to this endoplasmic reticulum is cis phase. Then we have this pouch like thing, this is your cisternae, and this one is known as trans phase or maturing phase, and each contains specific enzymatic functions. Protein and lipid synthesis in the endoplasmic reticulum enter the Golgi at the cis phase, undergo modification as they pass through the medial cisternae, and are sorted and packaged at the trans phase for delivery. So, these are basically help in the transportation system of any cell. The Golgi packages molecules into membrane bound vesicles, then destined for lysosomes and plasma membrane or secretion outside the cell. So, actually the packages, they are like for transportation we require packaging. So, this Golgi apparatus packages these important molecules or proteins or lipids into membrane bound vesicles, and these vesicles are travel to for lysosome, and which will be travel to the site of lysosome or to the plasma membrane or the secretion outside the cell, depending upon the requirement of the cell. It is involved in the synthesis of complex polysaccharides, which actually in plant cells, because the polysaccharides are not available in our body, and various glycolipids and sphingomyelin. Sphingomyelin means these glycolipids are lipids, and the sphingomyelin means they are one type of complex which are helping or which covers the nerve cells. The Golgi apparatus plays a key role in the formation of lysosomes and proteoglycans by attaching protein to the glycosaminoglycans. So vesicles boarding from this Golgi transport their cargo to the final cellular destination and ensure accurate intracellular trafficking. The Golgi apparatus supports cell secretion, membrane composition maintenance, and extracellular matrix production. These 3 are also important from the cellular physiology. Next one is your lysosome. Next important one is lysosome, and this lysosome are membrane bound spherical organelles. They contains hydrolytic enzymes responsible for intracellular digestion and recycling of cellular waste. So this lysosome, actually they are also known as very much useful for digestion, intracellular digestion, and the lysosome, it is also known as a suicidal bag or sac. The lysosomal lumen maintain an acidic environment which is in between 4.5 to 5 the pH and optimal for enzyme activity. It isolates from the neutral cytosol, because cytosol is neutral, by a single phospholipid bilayer membrane. They digest biomolecules such as proteins, lipids, carbohydrates and nucleic acid, breaking down them into reusable components. Lysosome participate in autophagy and engulfing and recycling damaged organelles and macromolecules generated inside the cell during this metabolism process. So this is also known as suicidal sac or suicidal bag. Lysosomal enzymes are synthesized in the rough endoplasmic reticulum, matured in the Golgi apparatus before being delivered to the lysosome through vesicles. They also play a role in cell signaling, plasma membrane repair and programmed cell death called apoptosis. Like that, mitochondria also helps in apoptosis. We have already seen that in the previous slides. Lysosomal membrane proteins protect the cell membrane from digestive enzymes and regulates import and export materials. So these are some about your lysosomes. Then another element or organelle, it is peroxisome. This peroxisome are small. They are membrane bound organelles that contains oxidase enzymes which catalyze the breakdown of fatty acids and amino acids via oxidation. So we have seen some of the in the previous case we have seen this lysosome contains hydrolytic enzymes responsible for intracellular digestion like carbohydrate, proteins, lipids, nucleic acid breakdown, all these things. And here you mark the peroxisomes, they contains the oxidase enzymes which catalyze the breakdown of fatty acids and amino acid by oxidation reactions. They detoxify harmful substances such as hydrogen peroxide H2O2. So this hydrogen peroxide H2O2 by converting it into water and oxygen using catalase. Peroxisomes plays important role in lipid metabolism, including synthesis of plasmalogens, key component of myelin sheath in nerve cells. They engage in the beta oxidation of very long chain fatty acids which mitochondria cannot process. So very long chain fatty acids mark the exception. All other things can be oxidized in a mitochondria and energy will be produced, but peroxisome is helping or engaging in the beta oxidation of very long chain fatty acids which mitochondria cannot process. The peroxisome also participate in metabolism of reactive oxygen species, helping maintain cellular redox balance. Like lysosome, peroxisome originates from the endoplasmic reticulum and proliferated by growth and division. So we have completed all the components, major components or organelles present inside the cell. So the main function of living cell is synthesis of genetic material, energy production, adenosine triphosphate in form of adenosine triphosphate, growth, cell division, replacement of old cells by autopsy, and reproduction of for development of new organism. So, cell death, that is your apoptosis, is the phenomenon of programmed cell death. When a cell dies on the external surface of the body, it is usually slugged off, but when a cell dies inside the body, it is ingested by phagocytic cells. Thank you. We'll learn about tissue in the next tutorial.